So today, we're doing a deep dive into polyploidy. It's a truly captivating biological phenomenon and actually far more common and powerful than you might first think.
Hmm.
Our mission for this deep dive is to unpack what polyploidy is, how organisms get it, and why it acts as this kind of hidden superpower for adaptation and evolution, especially in plants.
Yeah, and you'll discover some pretty surprising facts, like how it influences everything from a plant size, its structure, even its cell wall makeup.
Right, and maybe even how it could help us navigate, a changing climate.
To really get into that, let's first get a clear picture of what polyploidy actually is. Basically, it's the state where an organism has more than two complete sets of chromosomes.
Okay, so most sexually reproducing species, like us humans, we're diploid. We have two sets, like our 23 pairs of chromosomes, two full sets.
Exactly. So if N is a single set, a diploid is 2N. But polyploids, they take it further. They can be tetraploid with four sets, that's 4N.
Or hexaploid, 6N, octoploid, 8. It just keeps going.
Pretty much, yeah. And it's this ability to stack up chromosomes that makes it so incredibly prevalent, particularly in the plant world. I mean, well, you might find it sporadically in some animals.
It's everywhere in plants.
Astonishingly widespread. About half of all plant species alive today are thought to be recent polyploids. And even more remarkably, most modern seed plants. They actually have ancient polyploid ancestors. It's fundamental to plant evolution.
Wow, okay. That's a huge difference between plants and animals then.
Yeah.
And you mentioned there are different ways this happens based on where the extra chromosomes come from?
Precisely. There are two main types. First, you have autopolyploids. You can think of this as kind of self-doubling.
Okay.
All the extra chromosome sets come from within a single species. It's essentially multiplying that one species genome.
And the other type?
That's allopolyploids. These are quite different. They usually result from hybridization, a cross between two different species.
Ah, okay, like a hybrid plant.
Exactly. And then, crucially, there's a doubling of their combined genomes. What's really fascinating here is that this doubling often restores fertility.
So the hybrid might have been sterile otherwise?
Often, yes. Without that genetic duplication, the chromosomes from the two different parent species might not pair up properly during meiosis, making it unable to produce viable pollen or seeds. The doubling fixes that in many cases.
That's interesting. And just to be clear for everyone listening, this whole polyploidy thing, doubling entire sets of chromosomes, that's different from other chromosome issues, right? Like Down syndrome.
Absolutely. It's a really important distinction. Polyploidy is about whole sets 2N, 4N, 6N, conditions like monosomy, where you're missing just one chromosome, or trisomy, like trisomy 21 causing Down syndrome, where you have an extra single chromosome. Those are different. They're called aneuploidy, and they don't involve duplicating the entire genomic blueprint like polyploidy does.
Got it. Knowing those differences really helps you appreciate the sheer variety of ways life adapts and changes. Sometimes these big genetic shifts are where evolution finds its next major move. So we know what it is now.
Yeah.
But how? How do plants actually manage to duplicate their entire genome? You mentioned scientists used to think it was like somatic duplication, just cells making copies outside of reproduction.
Right. That was the prevailing thought for a long time. But that view has really shifted over the last few decades.
Okay.
It's now widely accepted that sexual polyploidization is the main route. And this happens through the formation of something called two-cell gametes.
Two-fin gametes. So gametes are usually haploid and right, like pollen or egg cells.
Exactly. They normally carry half the chromosome number. But 2N gametes, somehow, end up carrying the full diploid chromosome number instead. Two sets instead of one.
Whoa. So how does that meiosis mishap, how does that happen? What goes wrong in the cell division process, meiosis, to create these double-stuffed gametes?
It often boils down to something called meiotic restitution. Basically, specific errors happen during meiosis. One common mechanism involves problems with the cellular machinery, the spindles that guide chromosome separation.
The spindles.
Yeah.
Those are like the ropes pulling chromosomes apart.
Sort of, yeah.
Yeah.
Imagine the cell's internal GPS for sorting chromosomes gets, confused. The spindles might line up incorrectly, parallel instead of perpendicular in the second meiotic division. This messes up the final separation, leading to gametes that accidentally carry double the usual chromosome load.
Okay, so the chromosome sorting goes wrong. Are there other ways? You mentioned cell formation earlier.
Yes, that's another key area. Defects in cell plate formation. That's the new cell wall that normally forms after meiosis to separate the new cells. If that cell plate fails to form properly, either partially or completely, you can end up with cells containing multiple nuclei. These can then develop into diploid or even polyploid gametes. Sometimes the cell plate even forms too early before meiosis is totally finished, which also leads to these 2N spores.
And here's where it gets really interesting for me. You said stress can actually trigger this.
That's right. It's quite an intriguing finding. Research shows that abiotic stress, especially things like temperature swings, heat, or cold shock, can actually induce the formation of these 2N gametes.
So it's not just a random mistake?
It suggests it might not be just random error. It could actually be an adaptive response. The plant, when facing tough conditions, might increase the frequency of these meiotic mishaps. It's like stress increases the odds of a genetic shakeup happening right when the species might need new genetic combinations to survive.
Wow, okay. Like a built-in response to environmental pressure. Do we know what's happening at the molecular level? Like the genes involved?
Yeah, researchers are starting to pinpoint specific genes and proteins that act as, the regulators in all this. There are genes controlling how the spindles orient, proteins involved in building that cell plate correctly, and others that seem to oversee the overall ploidy level of the gametes.
So it's a complex, regulated process, even when it seems like an error.
Exactly. Understanding these molecular players really highlights how dynamic plant reproduction is, and how these seemingly small cellular events can trigger massive evolutionary consequences.
Which leads us nicely into the evolutionary impact. For a long time, polyploidy was kind of seen as an evolutionary dead end, wasn't it? Like a genetic abnormality that didn't really lead anywhere useful.
Oh, absolutely. That perspective has completely flipped. It's now recognized as a major driver of speciation, the formation of new species and diversification across a huge range of plant groups, and even in some animals, too. The old view just drastically underestimated its power.
So what gives it that power? You hear terms like genomic buffering and plasticity. What does that actually mean for a plant that suddenly has, say, 4 sets of chromosomes instead of 2?
Well, think about it like having multiple copies of every blueprint. If one copy gets a small error, a mutation, you've got backups. That's the buffering effect. It can protect against harmful mutations having an immediate negative impact.
Okay, like a safety net.
Right. But it's more than just safety. Having all those extra gene copies dramatically increases the raw genetic variation available, and it also seems to facilitate what's called epigenetic remodeling.
Epigenetic, that's changes in how genes are used, not changes to the DNA sequence itself.
Exactly. Changes in gene expression patterns. Polyploidy seems to open the door for more of these epigenetic changes. All this together, the buffering, the variation, the epigenetic flexibility, gives the organism immense potential to adapt, to respond more quickly and effectively to new challenges or environments.
It really does sound like a kind of evolutionary superpower, especially for surviving tough times. Is there evidence linking polyploidy to big historical events like mass extinctions?
There is, and it's quite compelling. If you look back at the fossil record, major ancient whole genome duplication events often line up, sometimes strikingly, with periods of dramatic global change and mass extinctions.
Like the one that wiped out the dinosaurs.
Precisely. The KPG boundary event, the Cretaceous-Paleogene extinction. There's evidence suggesting a surge in polyploidy among surviving plant lineages around that time. The idea is that plants with duplicated genomes might have had a better shot at surviving those cataclysmic environmental shifts.
Why? Because the stress triggered it, or because they were just better equipped?
Maybe both. The intense stress could have increased the rate of 2N gamete formation, and simultaneously, those newly formed polyploids, with their enhanced genetic toolkit, might have been better able to cope with the drastically altered conditions. We see hints of this at other major extinction boundaries, too, like increased levels of unreduced basically 2N pollen in the fossil record around the Triassic-Jurassic transition.
Fascinating. And this isn't just ancient history, right? We see this resilience in polyploids today.
Definitely. Polyploids are often found thriving in extreme or challenging environments right now. Their frequency tends to increase as you move away from the equator towards the poles into colder regions. They're common in dried areas, cold habitats, high altitudes. We have examples like certain Arabidopsis species showing increased salt tolerance compared to their diploid relatives. Polyploid versions of rice and citrange, a citrus relative, also show better tolerance to salt and drought.
And I think I read somewhere about aspen trees.
Yeah, that's another good example. Studies have shown that triploid aspen trees with 3 sets of chromosomes are significantly more drought resilient than their normal diploid counterparts.
That's pretty clear evidence. And it's not always the whole organism becoming polyploid, is it? Sometimes just parts of it.
Right, that's called endoduplication. This is where individual cells within an otherwise diploid plant become polyploid. It's seen as a plastic response, a flexible adaptation to various
Like
stresses.
what kind of stresses?
Things like intense UV radiation, low temperatures, drought, dealing with contaminants in the soil, fighting off diseases, even responding to physical wounding. By increasing the chromosome number just within specific cells, the plant can boost the size and gene copy number locally, allowing for a rapid, targeted response right where it's needed. It's another layer of adaptability.
And then for those allopolyploids, the ones formed from hybrids of different species, they get an extra boost, right? This hybrid vigor.
Exactly. They combine potentially advantageous genes from two different parent species. This can lead to heterosis or hybrid vigor, making them often more robust, faster growing, or more resilient than either parent species. Plus, they inherently have increased genetic diversity right from the start. It really stacks the deck in their favor for survival and success.
It really is captivating when you frame it like that. I mean, imagine a species effectively responding to something like rapid climate change by shuffling its entire genetic deck, getting a whole new hand to play with.
It's a powerful image, and it's not far off. It's happening naturally, and it's just this incredible testament to the adaptability and resilience built into life itself.
Okay, so polyploidy is clearly a potent survival strategy.
Yeah.
But how does it actually change the plant physically? You said it's more than just surviving. It can lead to profound transformations.
Absolutely. And this is where studies on model plants like Arabidopsis thaliana, the lab rat of the plant world, give us some really crucial insights.
What do they do?
Well, researchers create a series of Arabidopsis plants with different levels of polyploidy diploids, 2N, tetraploids 4N, hexaploids 6N, even octoploids 8N, and then they compared them very carefully.
And what kinds of differences did they find?
They saw some significant shifts in growth and morphology. Generally, the polyploids developed a bit slower than the diploids.
Slower, yeah.
Yeah. But interestingly, the tetraploids and hexaploids often grew into larger rosettes, that's the leafy base of the plant, and they had noticeably larger cells.
Bigger plants overall, then.
In many ways, yes. For instance, the tetraploids showed about a 20% increase in stem height compared to the diploids. And get this, an 82% increase in total dry weight.
Wow, 82%. That's huge.
It is. They also saw increases in the size of petals and sepals in all the polyploids, and increases in individual seed weight and the width of the seed pods, the calyx.
So it seems like polyploidy often leads to this gigas effect, just bigger structures. But is it always beneficial? Is there a downside to just getting bigger and bigger with more chromosome sets?
That's a really important question. And no, it's not always bigger is better. There's definitely a flip side. At very high ploidy levels, like the octoploids in that Arabidopsis study,
The 8 named ones.
right, they actually started showing structural problems. Things like distorted cells in the stem cortex, disruption in the central pith cells. Some cells even appeared crushed, and they saw collapsed vascular tissues, the plumbing of the plant.
So problems with transport.
Potentially, yes. And they also found a reduction in the thickness of the secondary cell walls in these octoploids, so it suggests there are definitely limits. Too much ploidy can lead to instability or structural weaknesses. It's not a simple linear relationship.
Okay, that makes sense. There are trade-offs. Now, you mentioned earlier the impact on cell walls, and this connects to something really relevant today. Biofuels. This feels like a potential aha moment.
It really is quite eye-opening. The research on Arabidopsis found this surprising correlation. Polyploidy in the stem tissue was negatively correlated with lignin and cellulose content.
Meaning less lignin and cellulose.
Less lignin and cellulose relative to other components, yes. But interestingly, it was positively correlated with matrix polysaccharides, things like hemicellulose and pectin, other types of structural sugars in the cell wall.
Okay. So why is that a big deal for biofuels?
Well, lignin especially is a major headache when you're trying to break down plant biomass to get sugars for fermentation into biofuels. It's like the concrete in the plant cell wall, really tough and rigid, and it blocks access to the cellulose.
Right, it makes it hard to process.
Exactly. So the finding that these polyploid Arabidopsis stems had altered cell wall composition, specifically less lignin relative to other polysaccharides, meant they were significantly easier to saccharify.
Saccharify, meaning easier to break down into sugars.
Precisely. They could be converted into fermentable sugars much more efficiently. This is potentially a huge deal for making biofuels more cost-effective and sustainable. If you can engineer or select for polyploid crops with this trait, you might need less harsh pre-treatment to release the sugars.
So this isn't just about plants adapting in the wild. It has direct, practical implications for how we might breed crops in the future. For energy.
That's the exciting prospect. These findings strongly suggest that inducing polyploidy could be a really valuable breeding strategy, not just for potentially increasing yield like we discussed with the size increases.
But also to specifically tailor the plant's biomass composition.
Exactly, tailoring it for specific uses, like making it inherently better suited for bioenergy production.
And this isn't just pie-in-the-sky stuff for the future, right? Aren't many of our current crops already polyploids?
Oh, absolutely. Many common crops we rely on every day have benefited from polyploidy. Think about potato, red clover, sugar beet, even seedless watermelon. Those are often induced polyploids. And some of the world's most important staple crops, like wheat.
Wheat is polyploid.
Red wheat is a hexaploid, 60M, and maize, or corn, has an ancient polyploid history. It went through a whole genome duplication millions of years ago. So, yeah, we're already heavily reliant on this phenomenon in agriculture.
So, understanding this biology isn't just academic. It has tangible connections to feeding the world and potentially powering it more sustainably.
That's the takeaway, yeah. It bridges fundamental biology with very real-world applications.
Okay, we've talked a lot about the advantages, the power of polyploidy. But let's bring in some nuance. It's important to understand it's not some kind of universal magic bullet, is it? It doesn't always work out.
That's a critical point to make, absolutely. While polyploidy can offer significant advantages, it's definitely not a guaranteed win. Many whole genome duplication events probably just fail.
Fail how?
Well, newly formed polyploids aren't always inherently better adapted than their diploid parents. They can face immediate challenges like genomic instability, maybe the duplicated chromosomes don't segregate properly during cell division. And meiosis itself, the process of making those gametes we talked about, can become much more complex and prone to errors with multiple chromosome sets trying to pair up.
So it can be a difficult start for a new polyploid lineage.
It can be, yes. And even if it survives initially, there's often a long process afterwards. You mentioned diploidization.
Right. What exactly is that?
So after a genome duplication event over evolutionary time, often millions of years, the polyploid genome tends to undergo changes that make it behave more like a diploid again. Chromosomes might get rearranged, genes get lost, and the duplicated gene copies can start to diverge, maybe taking on slightly different or even entirely new functions.
So the polyploid signature gradually fades in a way.
Functionally, yes, it becomes diploid-like in its behavior, even though it originated from duplication. This whole process means there can be a significant lag time between the initial polyploidy event and when its major evolutionary impacts, like bursts of diversification or key adaptations, become really apparent.
Interesting. So the benefits might not show up immediately. And we should also remember, polyploidy isn't the only way plants evolve and succeed, right?
Far from it. It's just one, albeit powerful, force among many.
Huh?
You only need to look at groups like the gymnosperms, conifers, cycads, ginkgo.
Pine trees and things like that.
Exactly. That's a hugely successful ancient lineage of plants dominating many ecosystems worldwide. And yet, polyploidy is remarkably rare among them. It clearly shows that massive evolutionary success doesn't always require doubling your entire genome. There are other paths.
So it's a much more complex genetic and evolutionary dance overall.
It really is. The consequences of polyploidy itself are multifaceted. It's not just about having more genes. It involves complex shifts in which genes get turned on or off, gene expression, changes in how genes are spliced together to make proteins, alternative splicing, even alterations in protein production that don't always neatly track the gene copy number. It's intricate.
A lot going on under the hood.
Definitely. And understanding this complexity, rather than looking for a simple story, actually gives us a richer, more accurate picture of how life diversifies and adapts.
Well, that brings us towards the end of our deep dive today into the world of polyploidy. We've journeyed through its fundamental cellular workings, explored its really profound impact on plant evolution and adaptation.
Right, how it acts as this engine for change.
Yeah, and even touched upon its quite surprising implications for our own future agriculture biofuels. It's really a testament, isn't it, to the incredible flexibility and resilience built into life's genetic code.
It really is. Thinking about how these sometimes massive genetic shifts, occasionally triggered by the environment itself, have shaped the plant kingdom we see today is just fascinating.
It definitely is. And it leaves us with a final thought, maybe something provocative for you listening. Given that polyploidy can be triggered by stress, and it seems to have played this crucial role in helping life weather past mass extinctions, what might that mean for us now in our current era of rapid climate change?
That's a powerful question.
Could we be perhaps on the cusp of another biological revolution. Maybe another explosion of polyploid species, either happening naturally out there or perhaps through more directed human breeding efforts, fundamentally reshaping the plant world as we know it.
It certainly gives us something interesting and maybe even a little urgent to think about.